So I would now like to go ahead and introduce Dr. Kerik Cox. Kerik manages a program of tree fruit and berry research and extension at Cornell Agritech, which is the New York State Agricultural Experiment Station. And his principal research efforts include antimicrobial resistance for both fungicides and antibiotics, and applied disease management with a focus on apples, stone fruits, and strawberries. Extension efforts focus on pesticide education, disease forecasting, and applied disease management, with emphasis on covered production in small fruits. And since the establishment of his program, Dr. Cox has been conducting antimicrobial resistance and invasive pathogen surveys in New York and the Northeastern United States. So looks good, Kerik, and you can go ahead and get started.
All right, thank you, Janet. Yeah, we're gonna go ahead and get started. In this talk, I have up to, the first thing I do is turn on that 40-minute timer, right? Let's do that. It's the most important part of the whole meeting. All right, staying on time is never a bad thing, and getting on the right slide set. So I have a couple of different things. One of the things, I'm probably not gonna spend a lot of time talking about best management practices, and we're gonna skip through a lot of those slides, mainly because I really want to get down to the sort of biologicals and the biopesticides and some of the other stuff. And I've probably talked a lot about the best management practices. So we're going to smooth through some of those. And for some reason, if I need to talk for longer, we can go back and visit those types of things. But I really want to make sure we really get down to sort of the nuts and bolts. Oh, we have a question? No, does not look like it. Good. All right, we're going to keep going. Then awesome. So with that, I'm going to kind of skip through a little bit of this really quickly. We can really talk about, and we all know the fire blight's bad, and this is where I'm going to focus my efforts. It's gotten really awful recently. We have our high density super spindle plantings, fairly expensive acreage, and trees get really long protracted bloom. It means they're going to be flowering a little bit later, particularly in the really early stages of establishment, on into the summer where the temperatures get hot. The models say spray every day. And on top of it, we need to hit that top wire. So we're going to need to make them somewhat vigorous. We can't completely arrest all of their development, or we'll never, ever have fruit ever again. And on top of that, yeah. And sometimes if you get a resistant rootstock, it's great idea. But if once the fire blight starts to look like the tree to the right of the slide, it's too late. And sadly, almost everything is susceptible, and we're getting a lot of really warm weather right at that petal fall period, which is actually, oddly enough, even though the flowers are going away, it's still a fairly susceptible time we're coming to find out for fire blight. And what's even worse about it is there's a lot of pressure to sort of stay away from the antibiotics. And so what I'm going to do is we're just going to skip through some of this stuff, and I really want to get to that next section. Yeah, I will just keep omitting them as we go through. Yeah, so we talk about this. This is where your biologicals are going to hit, and this blossom blight phase. Now, this is how the whole thing starts. The fire blight bacterium comes in and infects the blossoms, and in that particular instance, and then it will lead to a systemic infection that will blight the whole flower cluster. Usually, infections will happen around here, and by the time they start, forming little tiny fruitlets, that's when you start to see the symptoms. But at that point, it's racing through the trees, and this is where all of our biologicals, most of them will come in here at this particular stage of the game. You're in your PGRs and defense inducers and other various antimicrobials. And then when you get to the shoot blight, this is the worst part about it. This is where all the systemic infection is going to get in there and wipe out the entire tree. You're really going to be managing this with a combination of pruning, and labor is a little challenging for that one. And you have a defense inducers and various combinations of the PGR Prohexadione calcium. We'll be doing the most of the work in this phase. At this point, fire blight's inside, and it's just racing to the central leader as fast as it can. So, I mean, yeah, you can put copper and things out here to stop this, but it's really stopping this ooze from getting that shoot over there or the tree down the row. Once it's inside, it's sort of like on a slide to kill your tree for the most part. And I'm going to skip on ahead a little bit more. We'll talk some about this if we need to revisit it. Talk a little bit about some of the things you can do to put your best foot forward later with silver tip and where a lot of these things will come through. I'm going to sort of scoot on ahead a little bit. You can get horticultural, a little bit of resistance, but there's not a lot of single gene resistance in popular cultivars. And I mentioned there are resistant rootstocks, but once you get that fire blight into the central leader, you then effectively have a sprinkler system for fire blight in the middle of your orchard, and you probably need to move it. Yep, there's another picture of one of those such sprinkler systems right there. We're going to skip on through that, talk a little bit about cleaning up. A lot of these things you can do, and other talks that I've talked about are just going to really give you the heads up. If you do a good bit of winter pruning and look for things in the spring, we can skip through that. Don't forget your delayed dormant copper for both apple scab and fire blight. If things start oozing, it will help. All right, and then you're sort of moving through the system. I've been recommending some early Prohex to kind of like, we'll talk a little bit about that later, to sort of start the slowing down process now. And then when it warms up, letting the tree a little bit off the hook as it gets closer to terminal bud set. We'll sort of move through some of this stuff. And then the bloom. And this is where all the model alerts and the extension alerts and your regional program alerts are going to be more, even probably more useful than looking at the models, because they'll have experts running through every possible scenario. And we'll sort of skip through that. I mean, basically, if they're really, in most cases, seasons, you really only need one well-timed application. You just, unlike me, you don't know when the fire blight's going on. So that's where it gets a little tricky. And then common sense is good. We're leaving the realm of streptomycin because this is about biological tactics. And the end of antibiotics in organic happened a few years ago in 2014, and now there's a lot of biopesticides, defense inducers, and low MCE copper products, and these are starting to sort of taking the place in the organic fields. That, other than the fact that organic are often so low vigor that the level of fire blight they get is not that challenging to manage. We'll talk about this one when we start seeing some data. This is Biopesticides polulans. This is a highly effective biological control that works in Washington State, Oregon, Michigan. I don't even know about Pennsylvania yet, but it is working in every other program, including mine. It's very effective. It can be as effective as streptomycin, and we've been sort of leaning on that with the USDA germplasm repository for some of their collections as well. It's been a pretty nice material to have. And when you do these, and now we're sort of creeping into ideas about biological control, you may want to go out with these various, some will be protectants, some will be cidal, and we'll talk about which ones are which and as we go through the data. But and typically when we think of spraying an antibiotic, we start thinking of an EIP, and these are in the newer models. The EIP is present there. And like, look at my EIP over 70. In the case of a biological, I might need to start thinking about it, particularly if there's going to be wet weather. If it gets higher and it's a desert, you still need to be careful, but you want to start thinking with the biocontrol going for a lower EIP, which will mean, spraying more frequently, but maybe a little more costly. But it is possible to manage this disease with them, with the right amount of application skill and forecasting. Let's see, we'll talk a little bit about this. We're moving in the post bloom. We won't dwell on the standard program for Prohexadione calcium. Trying to avoid what I have in the picture over here by stopping the trees from growing and letting them grow again once they get into September. And we'll talk briefly about what that does, but I don't want to spend too much time on that. Copper's good. This isn't necessarily a biopesticide for the most part, but these are the types of things you can do as you begin to try to protect the surrounding trees. Think about copper as a protectant, and there are organic options. This is to protect that tree over there from fire blight on that tree. And always, if you're going to use copper, once fruit set is happening, seeing small fruits, you want good drying time. Otherwise, and a copper that mixes very well, and there are lower rates for post bloom. You want to hit those to really sort of keep that potential for spread down, but not rusted up your fruit. Okay, it is just a protectant, and the terminals will outgrow it, which means you have to apply it every week, and which means you have to risk your fruit finish every week. So it's something to be careful about. Talk pruning. Anytime you do anything with fire blight, you can start to see some of the maybe a little bit of ooze here and there on that nasty shoot. You want to do it on a cool, dry day. Any one of these things you do is going to really help get your biopesticides going. We'll sort of skip through a little bit of the rescue program and whatnot, and definitely stuff with the model, even though we sort of hinted on it earlier. And I really want to talk about the products. So we have our new model. Yep, I mentioned the EIP. You can find it in there as well. And you can sort of skip through this, or it can be shared later. And now I really want to get down to the real nuts and bolts. So let's talk about getting into these biopesticides and what's going on. Yep, I am going incredibly fast, Robert, because this is the actual part of the talk that you want to hear. Let me slow down when we get to this part. Okay, so what are these various biocontrol tactics? In terms of registration, there are 3 different types of groups, and biopesticide is the technical EPA definition term for these types of materials. And what they are is they're just naturally occurring derived pesticidal substance. They can be plant extracts. They can even be minerals. So for example, if you ever heard someone talk about putting on a phosphite fungicide, these are still biopesticides. They're not organic in every case, but they are biopesticides. Even microorganisms or their byproducts. Many of the things that are products that we use for fire blight are going to be the byproduct of a microorganism. The microorganism may be in there with it, but it's their naturally produced antibiotic that ends up doing the job. And then you might ask yourself, why can't we just purify that naturally produced antibiotic? And in the past we did, and that's how we got streptomycin and kasugamycin. But now, once we've purified them and mass produced them or synthetically produced them, now they're off the biopesticide list. If you play with them too much, they no longer become a biopesticide. Another good example would be some of the QOI fungicides, which if you could grind up mushrooms and smear them on a tree to prevent apple scab, you can use those strobilurins as a biopesticide as well. The other thing that not many people think about is that transgenes, things like BT, are actually considered biopesticides. Things like the honey sweet plum for plum pox virus resistance and stuff like that. So genes that we move around, I take the resistance gene out of topaz, I put it in the Macintosh, still technically a biopesticide. Would anyone ever buy it? Probably not. But neither here nor there. All right, so a lot of these things will be variable. There's so many different types. There's more types than I'll ever be able to cover here. We could spend hours talking about potentially different types of these things. And they cost as much as conventional products or more. They're a little hard to produce, when you're not synthetically producing the antibiotic and you're trying to make a perfectly balanced soup that has it in there. That also will impact the efficacy. These things, you're sort of relying on natural processes with a push from people to sort of make them effective products. The nice thing about them is that in many instances, they'll target multiple sites. So you don't have to sit there and think, am I going to get resistant to this biopesticide? A lot of times they're hitting a lot of different sites, which causes them to lack specificity and effectiveness. But at the same time, the chance of resistance is really low. At the same time, you might be thinking, well, couldn't I use one of these to help avoid resistance development? I'm like, yep, you sure could. And that's one of the things that we'll know. If you ever wanted to get a picture or a copy of the slide, you can click on these links and actually go check out the EPA list of biopesticides. And the thing that's most shocking is you'll be presented mostly with a big list of minerals at first until you scroll down a couple pages, and then you'll start to see the microorganisms. Okay, what else do we have? All right, now there's another group of products that are getting really popular, particularly in the Pacific Northwest, and these are FDA generally regarded as safe. Basically, these are food additives, things that you could put in foods and stuff like that, and they're safe enough for you to eat. It's become very popular as another tier above even organic. And a lot of essential oils and various oils will fall into this category as well. I'll talk about one that we test out. And then some things are just inert ingredients that are just so innocuous that they don't even need exemption. You have to watch out when you start to creep into this area. If it's not a labeled product, the company may or may not show efficacy data proving that it works. So it's something to think about. You can click on all these links if you had this talk and check out the various lists. I've just double checked them. We're not going to spend a lot of time fiddling with that at this point. So why would you want to use it? This is probably one of the bigger reasons. You get consumer acceptance and appeal. If you're growing organic, most of these things, now I mentioned the phosphite fungicides, they're not organic. They have to be naturally mined or naturally derived, and many of these biopesticides are. So that's a nice thing. If you have a production operation that protects against disease, you're not going to have covered production in apples, but it's entirely possible. If you have a small operation and you've got a good labor to size ratio where you can do a little bit of sanitation, these could be a good step for you. Yep. And anytime where you have frequent spraying possible, I already mentioned, spray every time the EIP is 60 or 70 and you have a rain event. You'll feel like you'll be putting on the biological controls quite often in that case. But if you have that agility, because everything is small, you can pull this off. Disease pressure is light. When you do these and you want to go organic, always start with various disease-resistant cultivars. And like there's not a lot for fire blight, and that's what we're going to talk about what to do instead. But the magic of organic is that sometimes the vigor is low enough that You don't get a lot of fire blight. Fire blight needs fast, actively growing tissues. So the more you slow the plant down, the less likely it's going to devastate the planting, which is why we're relying on that PGR apogee. And then finally, you need the right type of disease pathogen combination. You need something that's sitting on the surface, like this powdery mildew here, or maybe this fire blight, and it's fragile. Fire blight doesn't have a lot of protections against the environment. If I actually dump my fire blight in water and go out and try to inoculate my trees, there's a little ooze droplet right there. It all pops in the water and just explodes and it dies, and I get a terrible infection. Now, don't go spray your trees with water at bloom, trying to think I'll get fire blight with that. That will just end up in disaster. But for the most part, it is a very fragile organism, like this powdery mildew you see on this developing Ida red blossom cluster. These are the types of things you want to target. If it's deep inside the tissue, fire blight gets there later. It may not be the best approach for a biopesticide, but we can talk about some of the ones that can use. Now, is it the right disease for biocontrol? Yeah, we get these awful infections at bloom, and they lead to devastation, and they lead to these shoot blight epidemics. So at bloom, fire blight is this little fragile organism sitting on the surface of a flower. I would argue that it is probably the right one. But at the same time, preventing this blossom blight requires very precise applications. You might not think so. You think, well, I just go out to the model and I see when it's high risk and I apply it. That's the level of precision you need. And those days often occur when it's inconvenient. I know, because usually I'm sitting there waiting for fire blight to go, and it's always going to be the worst possible weather at, I don't know, Sunday morning or something awful like that is when the magic will really end up happening. But you got to go out and you got to do it. Typically, we've been using antibiotics, even to help with this precision. But there's been there's resistance developing in different regions. We have some here, but we're getting a handle on it because there are alternatives. And unfortunately, the antibiotics just lack consumer appeal. And every now and then I talk to someone in regulation, they're like, their time's coming, they'll leave. So I keep thinking, oh goodness, we need to do more stuff with biopesticides and figure something out. This pathogen is on the surface and it can dry out easily. Fire blight can withstand cold weather pretty well, but it cannot stand drying. And so we need something to just kill this and or dry it out. And but it's sort of limited and has no protective type structures. The ooze droplet is mildly protected in a bunch of sugar, but for the most part, it's possible to dry out. I mean, this can dry out and become like an amber, and then it'll stay there for a while and can be rehydrated. But in these early phases of those ooze droplets, the chance is there for these things to get it, if you will. So there's a lot of different modes of action. Here's some of the different diseases that I generally recommend for going after and biopesticides. Fire blight's one of them. Powdery mildew works really well. It's really popular to use this in greenhouse. This can be done in the field as well. There are a lot of options to pop these spores, which will also break in water. Fly speck sooty blotch is another particularly good one, particularly if you get a handle on the early initial inoculum at petal fall and first cover. And you might even be able to do rust in some instances. This one's kind of an aggressive pathogen. It can get away pretty quickly in an organic setting anytime you leave mancozeb, but it is potentially on the surface and something that could easily be killed. So there's 3 different types of modes of action of these things. One is they kill by the microbes producing some sort of metabolite. And this is your naturally occurring antibiotic that occurs in the soup. Now, if the team takes that antibiotic, purifies it, makes it highly effective, then it becomes another conventional antibiotic and it's off the table as far as a biopesticide is. And so that's one way. The second one, and then this one works the best for fire blight, we found, because we're trying to protect that flower stigmatic surface from getting colonized and growing and then getting washed into the cup. This is probably one of your better bets. The best antibiotic metabolite is an antibiotic. So almost everything that's a little less effective is going to have a little bit harder time getting rid of this unless it's purified and optimized for success. What's the final mode? The final one is defense inducers. This is kind of like the idea of let's ramp up the plant defenses, and can we stop this fire blight from getting deep in those floral tissues and moving into the tree and then killing our entire plant? For the most part. So that's what that's all about right there. We're all about slowing invasion and boosting plant defenses. But you could do all 3. You could kill, you could compete, and you could boost defenses. And we'll check out some of these things right now. For the most part, almost all the biologicals we're going to talk about are protectants. They're going to try to stop it from getting in, or are eradicants. It's like, oh, fire blight's there, I'm going to try to burn it out. When you think of eradicants, you think, I got to have those bacteria right there and my material on just at the right time. If there's any asynchronous attack in this thing, it's all going to be lost. Because in many instances, they have limited persistence. This isn't like a Captan that you need 2 inches of rain or 1 inch of rain to distribute and the second inch of rain to wash it off. These things go away fairly quickly, which is why they have to be applied often. Now, there's a lot of effort to sort of apply a lot of these different things with various adjuvants to try to lengthen their persistence and stuff as well. Oh, what do you know? Let's see if we can just mute that. Can't stop the phone, but I guess I can. How do I hang this thing up? Put it on mute. Let's do that button. There we go. That'll end it. Just to hang up. All right. So what are some different options that we got? These will be follow your double nickels. They'll be your protectants, eradicants, your serenades, your seraphiles, your tegros, your blossom protect, stuff like fractures, oils, and all the various essential oils are going to protect and eradicate, smother those cells. Good BM on that one. If it's Mike, I'll be looking around. Can they be combined with stuff like surround? That's a good question. It really depends. There's one thing that is important to note about combining things, as mentioned in the chat, is that when you start to combining them, there's a limited amount of surface area on the leaf for everyone. And so if some surround takes up that spot, then it won't be there for the other material. That's something to think about when combining. We did some work with combining various single sites for different diseases, and we found that stuff like powdery mildew will decline if a good powdery mildew fungicide is mixed with a good scablin, for example, as opposed to getting one or the other. So there's limited surface area for the leaf, and these things are on the surface. Okay, the second group, other than the few competitive inhibitors that we'll talk about, mostly it'll just be the Blossom Protect is about the only one in that category for the most part. And these work internally. They turn on defenses and they can be sort of synthetic or even natural defense inducers, and include things like actigard. Now, this is a synthetic conventional material here. This is a version of a natural defense inducer that has been optimized and highly optimized for the most and sort of largest plant defense response that it could possibly get. And from there, you also have some natural ones like Lifeguard, Regalia. Employ actually uses a protein from the fire blight organism to induce a defense response. And then we have Vaxaplant. And what's oddly enough is that even Apogee, according to some recent studies out of Michigan State, seems to turn on some defenses as well as its standard PGR type role, and kind of give you the 1-2 punch. Yeah, you could send out the PDFs. Seems fine. Okay, so let's talk a little bit about, we're going to get in and we're going to see some data on some of these different things. And as we go through the graphs, since I've got time, I have 17 minutes, I will go ahead and tell you which each of the products are and what they're doing as we look at them. So this is sort of our big overview of fire blight management. Very important, and always putting your best foot forward with the biopesticides is take those cankers out. Take the, and then come in with the copper. Anytime there's like a bud scale or a crack or any silver tip oozing, you're going to sort of just cover that and keep all the other tissues from getting potentially attacked later by oozing fire blight bacteria. Sort of smother it with that. And in some instances, at least in Geneva, we even go into 1/2 inch green a little bit with our copper stuff. And I don't think we've had too many issues, but we'll just leave it there sort of at the green tip for now. And when the antibiotics, which we won't talk about, but the biologicals, you're going to kind of go for the antimicrobials and protectant ones as soon as the flowers open. And with all the recent problems we've been having at petal fall, I'd say really make sure to get that first couple of days of petal fall until you're hit 100%. I'd say make sure to keep up pretty vigilant with your programs at that point. Now, when we get to the defense inducers, which I've labeled as SARS, which stands for systemic acquired resistance, there are a lot of different pathways in the resistance induced world, but one of the most frequent ones you'll hear about is the systemic one. You get an induction on one part of the plant, it leads to a signal that gets transported everywhere and boosts defenses for many days throughout the entire plant, which is kind of a nice idea. But bear in mind the defenses aren't not as strong as our immune system, but they can be pretty good. Are they good enough is the question. And then we talk a little bit about putting on Prohex and sort of the standard pedifall program when those shoots are starting to grow. Or you can even think about going on a little bit earlier and trying to stunt out these pedicels so they don't get attacked. But it's usually a season-long effort to do these things. And let's talk a little bit about what we got. So we've done a lot of different trials at Agritech, some on new plantings, which die very quickly, as you've seen in some of the pictures earlier of some of our high-density stuff looking dead. Yeah, that's just a loss. These nice bigger trees, the sort of standard vertical axe trees, on B9 that's one thing that is nice about these resistant rootstock is it allows us to do fire blight trials every year. I can decimate this tree one year, come back to the next year, and you won't even find fire blight on it unless I put it on there. Pretty nice, wide spacing. We can keep all the treatments away from each other. We can keep the fire blight from running all around the place, etc. And we do a lot on that. So a lot of times when it comes to certain biopesticides like the defense inducers, you want to try to ramp up the defense early for the most part. And in that particular instance, when you ramp up the defense early, you might go pre-bloom. And this is why we started poking at the idea of the PGR pre-bloom. It's like, let's just get stuff turned on. Let's get that slowing mechanism now before it gets too late and you get past bloom. And then for the most part, a lot of the antibiotics, I always try to get any company who's recommending an antibiotic or biopesticide to put their key product of interest on at 80% bloom. There's could be other applications early on to protect flowers, but this is mostly proof of concept for growers. My fire blight comes on at 80. So doing all the stuff ahead of time, there's really not a lot of natural fire blight out there. And for the most part, this is the big one. And so when I show a material in the graph, it had its application on right before the fire blight hit in our particular instance. You might ask, like, well, why don't you do one with natural inoculum? I did. The check usually has 0% fire blight in it. Interestingly, I don't even get fire blight in any of my blocks other than the ones I inoculate, which is kind of a strange thing, even though the rest of the Cornell Agritech experiment station could be melting. It's hard to say. Let's see.
What is that?
Oh, we got a little bit of good. We got a muter there. So we'll look at incidence of, I just look at incidence of how many percentage of all the flowers get blighted. And shoot blight, we either look at natural infection resulting from the blossom blights. For the most part, sometimes we'll actually inoculate, and this is where you can kill orchards very quickly. But for the most part, we do apogee, it could be pink, could be pedifall 1 to 2. This is sort of our standard for comparison. All the SARs, depending on the particular product, could go on as little as 2 to 5 days prior to infection. You want to give them a chance to sort of ramp up and see what they can do. In many instances, you never just use them all by themselves, but in our trials, I'll show them by themselves so you can kind of get an idea that, yeah, they can do something on their own. And then finally, the shoot blight, I usually look at how many shoots on an entire one of these monster trees gets fire blight, or I'll measure down the length and see how far it went. All right, so here's some data. We're going to start with some 2019 and we'll look at some 2020 data. The way to look at this thing is that, okay, about here, 100% of every blossom cluster I have has potentially blossom blight. This is the untreated check. Gets pretty high. And even on shoot blight on an untreated check, if we don't do anything, things really get killed. 60% of every first year shoot is just pretty much dying. It will make the tree look completely dead. And then, yes, we do lose some trees. You always have to throw in your antibiotic program for comparison, which is in orange. And the same with shoot blight. Even under my heavy inoculum trials, you still get a great level of control, even from a lot of different things. All these green ones are sort of be a lot of our biologicals. Some of them are a little bit weird, and they're sort of minerals. This alum is a pickling salt. And I suspect that it falls on there under the grass, generally regarded as safe category. And so that's what that works. This is your Blossom Protect. And this is your competitive blossom colonizer. Now, one of the potential caveats of this particular material that, we're not going to be talking the whole time about these, but I would talk about it later, is that the organism in this can cause russet. So it'd be important to sort of take care, but it's really not going to be used when the flower is exposed or even at petal fall. You want to stop this one before petal fall and mostly go up until late bloom. It has been known to cause russet. I've not seen it in the current formulations to do it, but I know the microorganism can do it. The other thing that watch out for this one is it is a really fantastic material. but it's a fungus. And so if you put on a lot of heavy fungicides afterwards, you might think, well, that didn't work. And mostly because your other cover spray killed it. And when you start thinking about mixing, this one is a really good material, but you probably almost want to go out and do it by itself. Serenade opti is another bacterium, and it makes a soup with an antimicrobial metabolite. This is Serenade opti. Oh, look, a little bit of actigard in there. That's a defense inducer. These two seem to be able to be mixed okay. This is double nickel, another bacterium in a soup. This is clava. This is a little bit of copper. This is Stargus, another antimicrobial, and this is a weird one down here. It's a bacteriophage. There are viruses that kill bacteria, and this is an attempt to make one that just goes after fire blight. It's a little more challenging. You have to do it quite often compared to some of these others, which really sort of went on maybe once or twice only. There are a lot of applications, but you can get some good control. What does it look like? So what do we say right now? This is for a pretty good one here with Blossom and Buffer Protect. It does seem to do a little bit of help to add that defense inducer in with your sort of bacterial soup biological, such as the Serenade product. What we got in 2020, slightly different year. There's the check again, nearly everything dead. Must have been a harder year for fire blight because I'm seeing a little more creep up in the old strep. Anytime you see a bunch of products being compared, you always want to see, did the check do really, really bad? And did the streptomycin do really, really, really good, or other antibiotic if it's a strep resistant area? And then from the others, you sort of just feed them in and compare and contrast. Let's see, this year we don't have too many different things. What do we have in this one? We have a little bit of employ. This one probably should be moved to the defense inducer category, but because that's the protein that induces defenses. But we left it there now. Here's an essential oil, a cinnamon oil. You can get control out of these, but 30% blossom blight is pretty rough. However, these aren't all used in a season-long program. And if this was sort of grower levels of inoculum, chances are you could do quite well with a lot of these things. I had a really good luck on agrophage. Whatever it was doing to sort of shut down blossom blight here seemed to carry on into later into the crop. But they can be successful, even in sort of a high inoculum trial. But 30% of all the shoots being dead is, or even 20, is a pretty ugly looking tree and a lot to cut out. But this is just for comparisons. You just want to compare this to that and that, and then you can get an idea, because even strep under this trial is kind of compressed. So we did pretty good luck. That's a really good product, and we've been using it a lot. Let's talk about some different defense inducers. Same sort of scenario. Really high on the check is what you want. Really low on the strep is pretty good, particularly with the shoot blight. This 2019 didn't seem like a very hard year. Lifeguard looking pretty good. Many instances, if the letters don't overlap, they're not statistically different. One of the things that you will find, let's see with this one, yeah. One thing to look at when you also evaluate these different things is look at the size of this thing. This is called an error bar. That tells you how much variability I have between the different reps of this double nickel clava one. And when you start looking at antibiotics and stuff, you'll see that there's a lot less variability. And when I mentioned that statement, like, hey, they vary a lot, this is what it is. The tighter the error bar, the shorter they are, the less variable they're going to work. You can see that even Blossom and Buffer Protect still has a really tiny one there, even on the shoot blight and on the blossom blight, indicating that it's been fairly consistent in how it performs. Well, they're not all bad. When you need to really concern is when the error bar looks like 50 and 20 or some of those. And those are types of things you can see. We did some regalia and added a little bit of defense inducer in there. We didn't see the synergy that other states had seen in that one particular year. What did we notice in 2020? We had some different things here. Similar high levels there. Like it's a worse year. We've noticed that. Had a phenomenal year for Lifeguard. Don't quite know why, but it was just very consistent. We've triple checked it every time. I did half the reps and Anna Wallace did the other half the reps. So who knows? Just worked out. What can you say? And then sometimes you'll see products really perform phenomenally one year and maybe just okay or pretty decent other years. And so this is it. Tight bars, short bars there. What else? We have some Vaxiplant. Cassumin, I had to put Cassumin on the mix because the Cassumin application went in at the 80. And that's one of the things I was talking about before. And that's why you'll sort of see it here as included with the Vaxiplant. I need to know a little bit more about how that one's working by itself before we can really compare it with the others. Might not have been a good year for Cassumin or Fire Lane, which is Oxytet. So that's Kasugamycin in there. And it could just been that it's a sunny year. The kasugamycin can diminish in the sun. And sometimes the oxytec can be challenging to make work. So it looked like they were trying to go for a different sort of punch. I mentioned that one. Yeah, Lifeguard did really well that year. What can we think about these? What can we takeaways? Why was Lifeguard less effective with copper? Let's see. Lifeguard with copper. That's a good question. And it could have been that could just been luck of the draw. It could have been that the copper went on closer to 80% bloom. It could have been that the two products interfered with each other. That's a good question, Glenn. Yeah, it's a weird one. In theory, I've gotten really good results with Quava by themselves. And then maybe this is sort of a cautionary tale of mixing. Maybe you get a little bit less of each and it's just not enough to do it, or they're incompatible. Let's see. One of the things I've seen is that Blossom Protect is incredibly effective. It is an organism that can russet. I've seen russeting from this mostly on stuff like, what is that, Miniesca or Sweet Tango. Never on the Argalas. Well, it looks like there's an extra M with agrophage. A lot of these agrophages, essential oils, and even pickling salts can be effective if you use them right. A lot of the bacillus products, really effective, and they can be enhanced with actigard. And as Glenn pointed out, we've seen some not so good combinations there as well. And maybe in that particular instance, a lot of these mixes may or may not work well in all situations. What about the defense inducers or systemic acquired resistance? There's Lifeguard, Regalia, even Vaxiplant, I suspect, even though there's some antibiotics in the mix, and Employ, the product Employ, can be effective at some levels, but they're not going to have the same wham aggressive punch as an antibiotic. And this is why I was mentioning, if you go at it with every other horticulture, every copper, every pruning, every other, and use your PGRs, you're going to have a good one. And from some new information that's coming out of Michigan State as part of our grant, it looks like you might get some of the strongest defense response out of actigard, but you're still seeing good control with the natural ones as well. So that causes you to wonder, like, why in many instances? This one may turn on the most genes, but it really depends. But across the board, a lot of states are finding actigard to be the stronger of the defense inducer materials. Aside from my program, which I get kind of a good level across the board from all of them for the most part. Do I want to talk about this? What is our time left? See, I feel like 3 minutes and 42 seconds. Yeah, we can creep a little bit. I didn't talk much about the PGR, so we'll sort of go into them a little bit. One of the ideas that we had is that it seems that in many instances, no one ever gets blossom blight. You don't see it. But suddenly you got this shoot blight coming out of nowhere. But what is hypothesized that there's 3 or 4 blossoms in the entire planting that somehow get blossom blight and then lead to this mess that you see on the right side of the screen. And it really sort of depends on, like, what's the host susceptibility, what's the level of vigor, and that will influence the level of devastation. I come to realize after doing a couple of trials at other places that we over-fertilize and our vigor is crazy. And sometimes the level of devastation I see is wild. And in that particular instance, we started thinking, like, wait a minute, we know that Prohex can slow EA down, but it also slows down young trees. And so one of the ideas that we thought about is, could we start going even before we even get blossom blight and try to slow down some of the pedicels so that we can stop this before the shoots even start growing and then let them grow out in September. And this was an idea that some greenhouse work on shoots in Michigan, and they were looking at, look at this, it's sort of thickening that cell wall. And then the idea is that this thickens, it's so thick that the bacteria can't pop that cell and inject the stuff it needs to invade. And we thought, like, what if we start putting it on these things, these pedicels? Could we stop the bacteria from invading? And, you know, we did some work with these sort of things. The idea being is stop it now before this creeps in there and starts rotting those shoots. And we did a little bit of work on this, and I'll just show a couple slides. I've shown it many times before. We did this in 5 years with Anna Wallace and myself, and looking at sort of these earlier applications, and we'll sort of end on that. One of the interesting things about this, and you can use the Prohex with the old actigard and kind of get a double effect in some cases. Really just depends on the year. Sometimes it can be better, and it's often better for the shoot blight. It can really creep down there even compared to a low. So what we have is the same sort of thing. I killed the trees. Strep looks great. Awesome. What about, there's the standard program of strep. It actually looks a little bit worse than just doing strep by itself. And you can start to see these come through with various noise. If I put them on a tight cluster to really see what happened, I tended to get a slightly smaller, maybe, it's literally highly variable level of blossom blight control than when I went on with pink. And then this one is sort of pink with a little bit of actigard at lower rates than all of them. It's like a 2 and 1 ounce in that particular instance, but it can look pretty good on the old on the shoot blight later, which is kind of what you're wanting to avoid anyway. So it's not a bad combo. And now we know it also turns on plant defenses. And very similarly, we did this in 2020, see a little bit more depending on the year. As mentioned, it was a harder year. Yeah, we started seeing more blossom blight protection. We went at tight cluster and more with a higher rate. So we sort of went up with those sort of higher rates in that particular instance. In this, this actigard one isn't as strong against the bloom phase of it, but really shines is getting that shoot light, which is what you want. And that's a combo that came out of one of George Sundin's students in Michigan, that sort of that two Prohex, 1 actigard. Not a bad, you go in sort of twice with it, once at pink, once at petal fall, to kind of go at that. Yep, and indeed you can really extend the thickness of cell walls in the pedicel using the apogee. And we did this at some commercial orchards. And the big takeaway home from this is that even if you have some young plantings, you can kind of go in with this stuff. And even in a wet year, it seems like the worst or the most impact you get is when you include it with a big full program of Prohex going twice at petal fall and 14 days later. These things don't have too much of an impact. And we did it in a couple different years. But for the most part, these are things you can do. Pairing with actigard is a pretty nice combination. Sometimes this is a practice for reducing bitter pit and honeycrisp. I've heard Mario often talk about pink applications of Prohex. And if you're doing it for that, you don't need to do it again for fire blight. And I'll go ahead and end my talk unless we can take some questions or move on to the next speaker. Yeah, they're ready.
Great. Thanks, Kerik. We have about 30 seconds left for questions. There was one, I think maybe this was a misunderstanding. There was a question about buffer protect. I'm wondering if that might have meant blossom protect, unless there is something.
Oh, yeah, blossom and buffer protect come as a combination unit. The buffer protect is indeed is for pH to make sure that the organism has the best chance.
Okay, and that's included in the product.
And it's included with the product. You don't need to buy two. It comes as a combo.
Okay, perfect. Thanks. And so we're out of time for questions now, but if anybody else does have any questions, feel free to pop them in the chat because we do have 5 minutes at the end of the talk. If you can stick around, Kerik.